Good afternoon, ladies and gentlemen. Thank you very much for attending ANGL's presentation. I can see some friendly faces, some existing shareholders and investors, but I can see plenty of new faces. So what I'm hoping to do is to explain to you something which I consider to be tremendously exciting, which is to explain how the cancer diagnostic market is developing, how a multi-billion-dollar market is coming, and how ANGL plc specifically is positioned to be successful within that market. And I think you'll find that some of the information I share with you is quite dramatic. That's the normal disclaimer, but the other thing to note here is that ANGL is not only listed on the London Stock Exchange on AIM, but it is also listed on the New York Stock Exchange on the OTCQX under the ANPCY ticker. So what am I actually talking about? I'm talking about something called precision medicine. Now, precision medicine is about getting the right treatment to the right patient at the right time. And this is important because everybody's cancer is different. It's a complete amazing thing to think that three patients might all have breast cancer, and yet they don't have the same disease. They have a disease in the same part of their body, which is very, very different. Cancer is extremely heterogeneous, and that is why some patients are treated and they recover, and other patients are treated and they don't recover, because they have different diseases, but they're treated with a one-size-fits-all approach. And that is what precision medicine is all about. And you can see on the right-hand side there that the pharmaceutical companies are onto this. They are developing selective medicines which will work very well, but only with some of the patients. So that opens up a huge question. Who are the patients who will benefit from a particular drug? And who are the ones who would benefit better from another drug? That's the question that we have to address with precision medicine. And the way it's done right now is what's called a solid biopsy. And that is very familiar probably to all of us here. That's where the surgeon will cut out some of the cancer and take it away and study it in the laboratory. Or they'll use a fine needle and they'll suck out some of the tissue. So it's a physical excision, a cutting out of the tumour and taking it away and analysing it and looking at those cells, looking at the cancer cells to try and find out for that particular patient, what is it that they've got in their condition of cancer and how can that be treated? What are the pathways? How does that cancer operate in such a way that it can avoid the immune system and grow rapidly? So that's all fine and dandy. That's how we do our solid biopsies when patients come and get treated. Obviously, some cancers are quite difficult to get in to cut it out, so that causes a problem at presentation. But even those which are easy to access, for example, the breast cancer, if there's a lump, it's quite easy to cut the lump out, and obviously the patient might well have a lumpectomy or a mastectomy afterwards. What if you want to check the cancer later in the patient's treatment? Actually, why would you even want to do that? Once you've checked it once, why do you have to check it again? The reason is cancer mutates. That's how it avoids the immune system. That's how it goes on and spreads. It changes all the time. So taking a snapshot picture when the patient comes into the hospital and then basing all the rest of their treatment on that picture is a completely false way of going about it, because the cancer will change. It won't stay the same as it was when it first arrived. And so it's absolutely crucial to try and understand what's happening with the cancer as it develops. But unfortunately, we don't do that now. And that's why we hear stories about friends who've got cancer and their disease has reduced by 90%. It's much, much smaller, and they look like they're getting better. And then unfortunately, we suddenly find out they're not getting better, they're getting worse. And that's because the remaining 10% of the cancer, the bit that hasn't been treated by the chemotherapy, is still there and it's growing, but it's different from the 90% and we don't know how to treat it. So what we need to do is be able to repeat biopsies as often as we want during treatment. But you can't do that because you've already cut it out. So how can you cut the same thing out twice? You can't. So what is coming is liquid biopsy. Now this is a phenomenon which is going to be a multi-billion-dollar market. It's going to bring precision medicine to the treatment of cancer patients, and it's going to change the whole way that cancer is treated. Because liquid biopsy involves getting tissue from the blood, from the patient blood, just from a blood test. And that is what our company, ANGL, is doing. And we have got something which is really, really clever. It's incredibly simple. but it's really, really clever. And this is it. That is a piece of plastic that's going to save a lot of people's lives in the future. It is a microscope slide shaped piece of plastic, and the blood will flow inside this piece of plastic. It's a microfluidic system which is designed to capture the cancer cells in the blood. Now, there's very few cancer cells, and that's why no one else has been able to do it beforehand. There's only one cancer cell in 1,000 million blood cells, so incredibly difficult to get hold of. But they are larger than the white blood cells and red blood cells, and they're less compressible. And this system, which has been developed by AGL over the last 10 years, is able to capture them. And I'm going to show you how here. This is a little animation showing you how the blood flows inside our cassette. It comes in through that inlet and it flows out of that outlet. So this is showing you the blood. It's an animation showing you the blood going through the cassette. And so what we see there is a series of steps. This is fully patented, by the way, and the core patent is in the use of these steps to separate cells or other particles in a liquid. So the cancer cells here are shown as green. They're much larger. They can't get over the critical gap. They get caught inside the cassette. But all the blood, other than the odd white blood cell and the odd red blood cell, get caught within the device. It's very, very simple. It's effectively a microfluidic filter. So what does that actually look like in practice? Now, this is a real-life cancer patient's blood going through our system. It's been photographed using a video camera on the end of a microscope at 100 times magnification. So what you see there in the deep red is actually multiple layers of red and white blood cells. And here, where it's much lighter coloured, that's where the blood is streaming through the critical gap in our device. So now you can see the blood flowing. Beautiful thing about it is it flows really easily through, doesn't clog up, no problems whatsoever. And it's streaming over those lines, and those lines are on top of the steps. If you look down on top of a staircase, you'd see the edges of the steps. So it's going up over the steps, and then the cancer cells are caught and everything else washes through. And I'm sure you'll agree with me, this is a phenomenally simple idea, but with a dramatic impact of the ability to capture cancer cells when there's only one in 1,000 million within the bloodstream. So then the cancer cells sit on our critical step. That's the smallest step in the device. And what we then have here is an automated instrument, and you can come and have a look at this instrument. We've brought one with us here, stand 27. Really, really simple bit of kit here, about the same size as a laser printer. And it just enables, you can't really see extremely well with this lighting, but there's a blood sample here. It's a standard blood draw. So the patient just has a normal blood test, a tube of blood in a normal standard tube. You take the top off it and you put it on our instrument, and then it's automated. Our instrument will push the blood through one of these cassettes, capture the cancer cells, and then we can reverse the flow and take the cancer cells out for analysis. And the great thing here is we're not reinventing any wheels. They already do solid biopsies, don't they, in hospitals all the time. They've got all the analytical techniques in their lab already. But what we're doing is saying, here's the sample. You can do a blood test from your patient. You can find out from that anything you want, the same information that you could have found out from a solid biopsy. And that's why we think this is changing the market and allowing precision medicine in cancer care. Now, why do I think the market is coming towards AGL in this? And I'll tell you why. Because 3 of the top investment banks have written research notes about the liquid biopsy market in the last 3 or 4 months. And this was the first one that I got my hands on. There's an extract here from the Goldman Sachs Investment Bank report on liquid biopsy. And they tell us that in 2025, so about 9 years from now, there's going to be $14 billion market per annum in liquid biopsy, doing the kind of thing I've just described in the United States alone. So you can treble that for the global market. Since then, we've seen Piper Jaffray and JP Morgan coming out with competing numbers. JP Morgan think it's something like 20 billion US in the same time frame. Notably, Goldman Sachs did quite a good job of segmenting the 4 different types of things that you can do with these cells if you can get them out. And as you can see, the first one is diagnostic screening. So early detection of cancer. You shouldn't have cancer cells in your bloodstream. If you have, that's an indication that you have got cancer. So early detection of cancer is a major market opportunity. I already mentioned about trying to work out which drug will help the patient at a particular time in their treatment, and that's therapeutic selection. And we've got pharmaceutical companies from all over the world who are beginning to engage with our technology as they develop new drugs which are very effective but only work for some of the patients. Thirdly, minimal residual disease. When patients are being treated with chemotherapies, these are toxic, right? They're toxic agents that nobody would want to take a chemotherapy drug. The only reason they take it is because they've got cancer and they want to kill their cancer. So what they'd like to do, the doctors would like to do, is to stop treating patients just as soon as they possibly can. But how can they determine whether to stop treating the patients? Answer is minimal residual disease monitoring, looking to see whether there's any residual disease. But as I said, if you've already cut it out, what do you look for? Well, we think that the circulating tumour cells in the blood is a marker that can be looked at. And finally, post-treatment monitoring. This is an absolutely crucial area. Patients who've been treated and they go into remission, it's great news until you hear they've relapsed, and then it's normally too late. So what tests are there to check that somebody who's had breast cancer, for example, and is in remission has not actually relapsed? Answer, nothing. There's nothing useful to do. They check for symptoms, and by the time the symptoms have turned up, it is too late. So you could monitor the blood for that. So anyway, Goldman Sachs and JP Morgan and these other chaps have all said there's going to be a big market, and that market is coming. The question is, what's it going to be composed of? Well, they've said it's the circulating tumour cells, so these intact cells that we capture, are actually the thing you really want. But it is too difficult to get hold of them. Wrong, Goldman Sachs. You haven't actually done your homework. AGL can get hold of them and can get hold of them routinely and easily. So that's why I think we're going to make a major difference in this marketplace. And in particular, there are, if you compare the solid and the liquid biopsies, you've got an advantage with the liquid biopsy where you can take a blood test as often as you want, it's non-invasive, and it's repeatable. When you look at the difference between ctDNA and CTCs, ctDNA is in relation, and when that's what everybody else thinks is going to be the main part of the market, that is looking at dead cancer cells which break up and they're in the bloodstream for a short period of time before they're excreted. The half-life is 15 minutes. So if you look at a dead bit of cancer cell, you can find out some information on DNA, but if you can get a whole live cancer cell which is in the process of trying to metastasize and cause a secondary cancer, you've got clinically relevant information, all the DNA information, all the RNA information, all the protein information, the ability to culture the cell and grow it, and maybe also use it in xenograft models with mice. So there are tremendous advantages over using CTCs as opposed to ctDNA. That said, AGL's strategy is not to try to compete with the ctDNA approach. Instead, we've proved that our system is completely compatible with it. You can use a single blood sample and you can extract the ctDNA for analysis, which is what the other people want to do with the DNA fragments. Their waste product that they would throw away, we can run through our system and get the actual intact cancer cells. So the two things are entirely compatible. Another absolutely crucial advantage of AGL's system compared to other systems which are being developed is that it is patented. We have a patent-protected system, and that means we can sell our instrument to hospital labs so they can do this analysis. It's a razor blade model. We sell them an instrument, and then we will sell them consumables, individual cassettes to be used each time a patient comes. The ctDNA and our competition in CTCs are stuck doing service lab approach. They have to have a blood sample sent to them, which they have to analyse. That's not what the hospitals want. Hospitals want to do the investigation in their own laboratory. And what's more, it's not a good commercial model. If you double the size of your business with a patented product like ours, we can do that instantaneously by telling our manufacturers to manufacture more product. But with a service lab, you're highly restricted. So we've got 7 key differentiators of our system compared to all of the other circulating tumour cell systems. But I want to move on and just tell you a couple of other key things before my time's up. Research use sales. AGL has started making research use sales, announced in December of last year, so just a month and a half ago. We've got multiple customers buying this for use in research. We've sold instruments and we've sold consumable cassettes, both to brand new customers and indeed also to other key opinion leaders who are transitioning across to become paying customers. There's a large market in research, £250 million per annum opportunity for us, and our pipeline is growing extremely fast. In terms of the absolute objective for Angle is to sell into clinical cells. That means for treating patients. That's the whole purpose of this. We have to have two things. We have to have patient data and we have to have regulatory authorisation. And I'm going to show you those two things, and then that's going to be the end of the presentation. But I would like people to come to our stand at 27 to discuss this. So FDA authorisation. We want to be the first company ever authorised for capturing circulating tumour cells, cancer cells, from patient blood for analysis. We've been in detailed discussions with the FDA for over 2 years now. We have a completely developed plan which we've discussed with them to meet their remaining requirements. We're focused initially on metastatic breast cancer, but this product is going to be across all the different cancers in due course. And we have 3 top US cancer centres lining up to undertake the final studies, and we're expecting to see some pretty strong progress this year on that. Our first clinical application is in ovarian cancer. That's an absolutely crying unmet medical need. 500,000 women a year have operations for abnormal pelvic mass, a growth which they're going to have surgery to have cut out. About 10% of them have ovarian cancer. It is shocking that there is no way of detecting ahead of their operation who the 10% are. We have developed a blood test using our system, as I described, with Medical University of Vienna, which can very accurately discriminate not only which women have ovarian cancer, but crucially, and other systems can't do this, it's specific. So it identifies those who do not have ovarian cancer correctly. Other systems incorrectly have false positives. We've got work going on with BARTS in prostate cancer. 52 out of 52 patients, we got cancer cells in prostate, including 8 early stage, which opens the opportunity for a detection of prostate cancer. We've got fantastic work with University of Southern California in metastatic breast cancer. So this is breast cancer that's spread. And they have already shown in a small number of patients that they can get the whole genome information from a blood test, which they would have had to get from surgical intervention. We're looking forward to the presentation by Julie Lang, hopefully at AACR, the big cancer conference in April, for the final results there. Lung cancer, Cancer Research UK, 12 out of 12 patients, massively outperformed the previous gold standard, as you can see there. Large numbers of patients where we detected epithelial cells and they didn't. They can't do mesenchymal cells, which are crucially important. And Jed Brady, who's a top person at Cancer Research UK Manchester Institute, was quoted as saying the Parsortix system, owned by Angle, has a unique combination of features, making it suitable for routine clinical analysis of patient blood samples. So this is it. What I wanted to say is this market is coming. It's a massive medical market. It's going to make a difference to a lot of people's lives that we know. And it's a UK company that's driving this technology, which is Angle. We've proven high performance in this system in ovarian, breast and lung cancers. We've got research use sales now, so we're generating revenues and it's growing fast. We're already CE marked, regulatory approved in Europe. And we've got an FDA authorization in process. Our first clinical application in ovarian cancer, the European study will be finished by the end of 2016. Thank you for your attention.